Fungal infections of the skin, hair and nail are also referred to as tinea, the Latin name for worm or ringworm, as some fungal infections cause a circular, scaly ring lesion on the skin. Examples of tinea infections include tinea pedis, which is fungal infection of the feet or athlete's foot, leading to discoloured, thickened toenails and scaling between the toes. Tinea capitis, which is fungal infection of the scalp, causing scaly lesions with hair loss. And tinea corporis, which is fungal infection of the body with scaly ringworm lesions on the skin. The main fungi that cause these infections are the dermatophytes, which are grouped into three genera: Trichophyton, Microsporum and Epidermophyton. They break down keratin in the skin and use it as a nutrient source, and the infections they cause are confined to the outer keratinized layers of the skin. The dermatophyte infections can either be zoophilic, associated with animal contact, geophilic, associated with environmental contact, or anthropophilic, associated with human contact. Some of the most common dermatophyte infections are Trichophyton rubrum, Trichophyton interdigitale, Trichophyton tonsurans, Epidermophyton flocosum, and Microsporum canis. The laboratory diagnosis of superficial fungal infections is made using microscopy and culture of samples of infected skin, hair, and nails. Samples are best collected into folded squares of black card or paper to allow drying out of the sample to reduce the level of bacterial contamination and also to allow easy visualization. This sample shows toenail clippings and scrapings. The card is carefully opened by the biomedical scientist wearing gloves and protective eyewear. Direct microscopy is performed using 20% potassium hydroxide on a slide, and fragments of the nail are added to the potassium hydroxide. A cover slip is applied and the slide heated on a hot plate for approximately 15 minutes to allow digestion and clearing of the keratin and skin cells by the potassium hydroxide. After digestion, the slide is examined under a microscope using the ×40 objective. Fungal elements are seen as branching hyphae running across the outside of the skin cells, as can be seen in this slide. Most labs now add a fluorescent stain called chalcofluor white to the potassium hydroxide preparation, with subsequent examination of the slide under a fluorescent microscope. Fungal elements will appear as bright yellow-green fluorescent hyphae as the chalcofluor white binds to the chitin in the fungal cell walls. Culture for dermatophytes is performed onto selective media called sabourode agar, which is a glucose peptone medium. Two types of sabouroodes or sab plates are available: sab plus chloramphenicol to prevent bacterial overgrowth and to grow non-dermatophyte fungi, and sab plus chloramphenicol plus actidione, which is also known as cycloheximide, to grow dermatophytes, but inhibit the growth of some non-dermatophytes that can be found as environmental contaminants, such as Aspergillus and Penicillium species. Samples are cut into small fragments and are inoculated using sterile forceps just below the surface of the media, placing at least 3 inocula onto each plate. The plates are then placed into plastic mini-grip bags to contain any fungal growth and any fungal spores that may be generated in order to prevent contamination of other plates. Plates are incubated at 28 degrees for at least 14 days with examination each week. On examination of cultures, identification of fungal growth is made by describing the growth, the colony morphology, is it flat, folded or submerged, the colony texture, is it floccose, waxy, granular or powdery, the colony surface colour, and the colour of the reverse pigment of the colony on the underside of the plate. This isolate from the toenail clippings shows a cream, flat, powdery colony with a brown cream reverse. To identify this isolate, a lactophenol cotton blue sellotape preparation needs to be made. A piece of sellotape is used and pressed onto the colony surface to pick up the fungal structures and spores. The sellotape is then transferred onto a few drops of lactophenol cotton blue on a glass slide. This preparation is then examined under a microscope using the ×40 objective. The slide is examined for the arrangement of the hyphae, the presence of small unicellular microconidia, or the presence of large multicellular macroconidia. This isolate shows small round clusters of microconidia and spiral hyphae, together with larger cigar-shaped macroconidia, and alongside its colonial appearance, it would be identified as Trichophyton interdigitale. To help identify fungi, the Health Protection Agency textbook, Identification of Pathogenic Fungi, is also often used. Trichophyton species are usually characterized by the presence of many microconidia, round or elliptical, with few thin-walled, smooth macroconidia. Microsporum species are usually characterized by the presence of many spindle-shaped macroconidia, which are often rough-walled and multi-celled, with few microconidia. Epidermophyton flocosum is characterized by the presence of small, clavate, club-shaped, smooth-walled macroconidia, with between 2 and 4 cells, and no microconidia. These culture plates on another sample show growth of 2 spreading colonies on the sabrodes plus chloramphenicol plate, but no growth on the sabrodes plus chloramphenicol plus actidione plate. Thus, this indicates that this growth is not a dermatophyte and is most likely an environmental contaminant or a non-dermatophyte, of which there are a few that can cause superficial skin infections. On the basis of fungal microscopy and culture results, the patient will be treated with topical antifungal agents, which are usually creams or powders, with occasional use of oral antifungals for more complicated infections.